Hi, my name is Bella and I'm excited to share with you the findings from my honours thesis, a psychophysical evaluation on subjective colour perception within the context of depression. The way we talk about depression, with metaphors such as the black dog, feeling blue, and the persistent presence of a metaphorical dark cloud, has encouraged scholars and clinicians to explore the interplay between psychological states and how we see the world around us. In this presentation, I will work on characterizing how colour is experienced by individuals with depression and examine the relationship between depressive symptoms and subjective colour experience. Major depressive disorder is a highly prevalent and devastating mood disorder, impacting approximately 5% of the adult global population every year. The disorder stands as a leading contributor to disease burden and disability worldwide. It also presents profound implications for biological, psychological, and behavioural well-being. Recent research suggests that depression may also be associated with various deficits in visual perception. Specifically, self-reported associations between a grey, cloudy, drab perception of colour and an increased severity of depressive symptoms have been observed, suggesting that our broad perception of colour may be altered by our psychological well-being. However, few studies have employed psychophysical tools to quantitatively investigate the relationship between depressive symptomatology and colour perception. Specifically, similarity relationships and the equivalency of colour experience have remained unexamined. Bridging this gap is the primary objective of my thesis. Depressive episodes have been linked to a reduced ability to distinguish between colours. In a recent study using the Farnsworth-Munswell 100 hue test, individuals experiencing a moderate to severe depressive episode, scoring 14 or above on Beck's depression inventory, exhibited a significant impairment in their capacity to discriminate and arrange colours sequentially. Another study by Pham et al. Employed subjective visual tests and revealed a negative correlation between the severity of depressive symptoms and the ability to discriminate visual contrast. This suggests that as depressive symptoms become more severe, individuals may experience difficulties in perceiving colours and contrast. Researchers studying depression have also observed differences in the central and peripheral visual systems. However, behavioural studies have mainly related to attentional bias. In a study led by DeZorzi, depressed individuals showed a central attentional bias, particularly for negative emotional stimuli. We can see in the graph presented here that depressed individuals demonstrate an opposite pattern of both sensitivity and reactivity across the visual field compared to their healthy controls. A recent study by Koska et al. Found that depressed individuals exhibited delayed central retinal signaling. This delay in central retina function may contribute to the dim perception reported by depressed individuals. The study also noted hyperactivity in the peripheral retina of depressed patients, possibly compensating for the central dysfunction. However, the exact purpose and functional effects of this peripheral hyperactivity remain unclear. This underscores the need for further research, which forms the basis of the aims of my study. Our study employs a novel colour dissimilarity paradigm, previously established by our lab, to explore the connection between depressive symptoms and colour dissimilarity relationships. Our first hypothesis is that higher levels of depressive symptoms will correlate with a duller perception of colour. This dullness is anticipated to manifest as both a lower mean and reduced variance in colour dissimilarity judgments. To illustrate this proposed difference in colour perception, I've included a graphical representation on the slide. If colours appear greyer or duller to participants, they are likely to rate them as less different from each other. This would then result in lower dissimilarity judgment scores and lower mean values. Even if individuals with high depression scores had the same average score as those with low depression scores, a duller perception would lead to less variability in their judgments, thus yielding a lower variance of scores. In addition to investigating colour perception's association with depressive symptoms, we aim to explore the equivalence of colour experiences across the visual field, specifically within a sample exhibiting high depressive symptoms. Our second hypothesis is that individuals with high depressive symptoms will report a duller perception in colour in the peripheral vision compared to central vision. We expect this hypothesis to be supported by lower mean and lower variance values in colour dissimilarity judgments in the periphery compared to the centre of the visual field. We recruited 500 participants via the online platform Pavlovia. We measured two main variables, colour dissimilarity perception and depressive symptomatology. Depressive symptomatology was assessed using the Beck Depression Inventory, known as the BDI, consisting of 21 statements related to depressive symptoms experienced in the past 2 weeks, with a total score derived from the sum of responses. We employed an online colour dissimilarity rating task adapted from prior lab research. This task featured 93 colours with variations in hue, saturation, and brightness, presented as solid coloured circles on a grey background at different visual field locations. Each participant rated 200 randomly selected colour pairs. To clarify the task, we've included a brief demonstration here. Participants view 2 randomly chosen stimuli displayed for 125 milliseconds. These stimuli can appear either in the centre of the screen or both in the periphery. Afterward, participants assess the dissimilarity of the 2 colours using a scale from -4, meaning least similar, to +4, meaning most similar. Lower scores indicate less perceived similarity between the colours. To ensure the reliability of participants' colour dissimilarity judgments, we implemented control measures, including attention checks and a double pass correlation. The double pass paradigm involved participants rating each colour pair twice for central and peripheral eccentricity configurations. The consistency of their responses could then be assessed by comparing both rounds. Now to discuss the results of our study. We divided participants into 2 groups based on their depression symptoms, one with low symptoms, scoring less than 14 on the BDI, and one with high symptoms, scoring 14 or above, indicating a mild to severe depressive episode. We employed a Mann-Whitney U-test to analyse the differences in mean colour dissimilarity scores between the 2 groups across the visual field, with our results revealing no significant differences. This held true whether we looked at their overall vision or separately examined their central and peripheral vision. We used the same test to analyse the variances of colour dissimilarity judgment scores between the groups of low and high depressive symptoms. Once again, our results revealed no significant differences. As neither the mean or variance of judgments differed significantly between the 2 groups, our first hypothesis was not supported. To assess our second hypothesis, we examined the colour dissimilarity judgment scores of participants in the high depressive group to assess the consistency of their colour perception across their visual field. We used a Wilcoxon-signed rank test to compare their mean colour dissimilarity judgment scores for central stimuli with those for peripheral stimuli, all within the same subjects. Results indicated no significant difference across the visual field. We also observed the same in the low depressive group. These matrices displayed observe the patterns of mean dissimilarity judgments across the visual fields of both groups and help us identify potential variations in responses for specific sets of colour pairs. Following our analysis and examination of the matrices, we found no significant differences in mean colour dissimilarity judgments across the visual field for either group. We conducted a Wilcoxon-signed rank test to compare colour dissimilarity judgment variances within the high depressive group, looking at central versus peripheral stimuli. Surprisingly, the high depressive group exhibited significantly higher variances in peripheral judgments compared to central judgments. This suggests a potentially unequal colour experience across the visual field, which was not observed in low depressive group. Refer to the matrices for a visual representation of these variance patterns for each colour pair. These unexpected results contradict our initial expectations and do not support our second hypothesis. Previous research has suggested a narrowing of the visual field in depression, possibly explaining the increased variance in peripheral judgments. Alternatively, the higher variance in peripheral judgments within the high depressive group might indicate heightened retinal signal activity. To confirm this relationship, future research should simultaneously assess retinal activity and subjective colour perception. Our research faced limitations in data collection. To enhance meaningful subgroup comparisons, future studies should consider larger sample sizes and ensure both depressive groups view the same colour sets, promoting direct comparability. Being cross-sectional, our study could not capture the direction or stability of the colour perception-depressive symptom relationship, and future research should explore past depressive episodes or employ longitudinal designs. Our online approach, while advantageous for recruiting a large sample, raised concerns about control measures and visual field analysis scope. Future research in controlled lab settings with standardized displays could extend retinal eccentricity, providing a more comprehensive analysis. In conclusion, our study marks the first effort to utilize psychophysical tools in characterizing and exploring the interplay between depressive symptoms and color dissimilarity relationships. Although our findings did not align with our initial hypotheses, they contribute significantly to the growing understanding of the complex relationship between depression and subjective colour perception. Our results raise questions about the reliability of colour perception deficits as an objective diagnostic tool for depression, underscoring the necessity for additional research examining the stability, retinal correlates, and metaphorical foundations of these potential deficits. Furthermore, our study lays the foundation for future research on subjective conscious experience within diverse psychological contexts, bridging the gap between psychological and perceptual experiences. Thus concludes my presentation. Thank you.